Welcome back, and today we will discuss again genetics in psychiatry. This was prompted by a recent paper in Biological Psychiatry, which found yet two more polymorphisms in the genome that are linked to major depression. Now here is the paper. It just appeared in Biological Psychiatry, and you can see by the number of authors that it's a mega study which did a genome-wide association meta-analyses to identify these new loci involving a truckload of patients. Now, first of all, I'd like to show you a brief video on the methodology and the explosion of findings involving genome regions implicated in disease traits and other traits since 2005. Now that you are back from this very brief video, let's continue our talk. Now, the finding in Biological Psychiatry was so extraordinary that it prompted a commentary by the editor of this volume. And the headline is quite revealing, namely, a genetic locus associated with depression, the iceberg begins to melt. Why iceberg? Well, in opposition to findings on schizophrenia, Alzheimer's disease, Parkinson's disease, and others, depression had been extremely recalcitrant and resistant in terms of linking specific loci in the genome to major depression. Now, this is not unusual because if we go back in the history, a brief history of genomic studies, it took about 8 years to move from early negative results in schizophrenia to more positive results, which culminated in the remarkable publication of 108 genome-wide significant loci in schizophrenia in the year 2014. The major reason for the failure to find anything was sample size. Now, in this 2014 publication by the Schizophrenia Working Group of the Psychiatric Genomics Consortium, more than 30,000 cases of persons with schizophrenia were contrasted to 100,000 controls. So you need substantial numbers to find these signals in the genome. Depression has been estimated to be 40 to 50% heritable, whereas schizophrenia is perhaps 80% or more heritable. So you can see that the odds are stacked against finding something in depression. There are other reasons too, which we will discuss later in this talk. So this then was the study that showed 108 schizophrenia-associated polymorphisms in the genome. Now, this was a breakthrough study for more than one reason. Number one, it pointed out the need for large numbers, and number two, it led the trail to a specific site in the genome, which then was picked up later and revealed a very interesting finding in schizophrenia research. So here it is. This is the Manhattan plot, which plots the likelihood of association here with a site on chromosome 6. And you can see here one outstanding finding on chromosome 6, which was later linked to a histocompatibility gene on chromosome 6, and it codes for a C4 complement gene. Now, why is this important, and why would this show up in schizophrenia? Nobody would have anticipated or expected that. And that exactly is the reason for these studies, not to find just a gene which accounts perhaps for 2 or 10% of the variance in the findings, but to find mechanism. If you find a gene, then you can track down associated genes and see whether you can identify a gene network which perhaps is operational and is involved mechanistically in the production of the disease. And here you see the C4 finding, namely, if you have a certain allele, it results in increased expression in the brain of the C4 complement haplotype. And that means that the C4 hyperexpression may be involved in some way in excessive synaptic pruning. I'd like to refer you to a video talk which is now included in a collection of talks called the Psychiatry Genetics Project, which will be accessible in a few days on our website. Now, going back to the history of depression. Despite many attempts, it was only in 2015 that for the first time significant association of depression and a locus in the genome was detected. And this was a special circumstance, namely, the patients in the study were from China. Now, in China, there is a certain bias and stigma towards mental illness, and patients don't go see a psychiatrist. Only the most severe cases, namely those involving perhaps melancholia and severe depression, may show up in the clinic or in the inpatient units in China. And this was the case with this experiment. So this study then looked at a large number of depressed patients in China, and positive findings were made in a gene called Srt1. And here is the title: Sparse whole genome sequencing identifies 2 loci for major depressive disorder. Now, sparse genome sequencing means just to take a quick peek and to run through the genome without many repeat runs to correct for errors, and relying instead on the large number of patients, washing out any inconsistencies and statistical variations. And the finding then was that with a very high probability, p of 10 to the minus 10, this locus here at the Crt1 gene was identified. Now, what does this gene do? Well, it is involved in the function of mitochondria, in the energy metabolism of the cell. Now, who would have suspected that depression has anything to do with mitochondrial energy function and the powerhouse that each cell has, providing it with sufficient ATP and power to conduct its business? And here is a comment: in East Asia, there is a reluctance to report major depressive disorder, and this probably explains why hospital ascertained cases are the most severe and why the prevalence of major depression in China is reported as being lower than in the US by about a factor of 4 to 5. It's unlikely that there is a 4 to 5 range in genetic predisposition. It is more likely that cultural and stigma-related issues are operational in this context. And so here is then the finding of the Crt region. You can see here again a classical Manhattan plot which identifies this one locus that survived a repeat analysis in a duplication sample. So again, we talked about mitochondrial biogenesis, and together with the finding that major depression is associated with increased amounts of mitochondrial DNA, suggests an unexpected origin of at least some of the phenotypic presentations of major depressive disorder. All right, so let's get back to the study at hand, namely the 2 genome-wide association meta-analyses that were found. In this case, 70,000 subjects were analyzed and the replication sample of 28,000. The initial sample to take a stab at this was 9,000 patients with depression and 9,000 control subjects. So this was supplemented with a population of 50,000 subjects who were assessed for depressive symptoms present a few weeks prior to the interview. So these were not folks that were diagnosed clinically by a psychiatrist using a standardized interview or standardized rating scales. Rather, any type of depression reported before the test was counted. And the finding then in the end will indicate that we don't need this sophisticated diagnostic identification of patients, but rather a more global report of depressive symptoms might be enough to fish out the genes that are important here. Now, these patients were then not diagnosed with DSM criteria and were not assessed for lifetime psychopathology. So here then are the results. There were 2 loci found, and the one of great interest is this one here, the Fhat gene. And here is the Manhattan plot, and you can see in no uncertain terms that this gene appears to be highly linked to depression in these gigantic samples. Now, there's only a single nucleotide polymorphism here in the Fhat genes that shows an association in the replication cohort of 28,000 subjects with major depression. And this was fished out by the meta-analysis. Now then we have 2 loci identified in linkage studies, namely severe depression in China with the Sirt1 gene, and here now the Fhat gene. Now it turns out that the amount of variance accounted by these loci is rather small. Now the Fhat gene, though, seems to be intriguing because it seems to be a circadian clock modifier gene and is related to increased daytime sleepiness, which may be salient for the diagnosis of major depression. So the Fhat gene is expressed in multiple brain regions, as you would expect for something involved in depression, namely the amygdala, anterior cingulate cortex, caudate prefrontal cortex, and hypothalamus. It is also assumed to play a role in oxidative stress and response to DNA damage. Both are biological processes that are implicated in major depressive disorder. So the conclusion then is from this study that a continuum of depressive symptoms and major depressive disorder may exist and yield a signal in the genome, which extends not just to severe major depressive disorder diagnosed by DSM, but rather to a population that reports any significant depressive symptoms whatsoever. So it appears that they are phenotypically more inclusive and thus may help achieve the large sample size, ideal to detect susceptibility loci for depression. Now, in the meantime, one more study identified more loci, and this is here. It appeared in Nature Genetics. And this was done by utilizing patients, or subjects rather, that participated in the 23andMe website. Now this is a site, as you may know, that offers you for a fee to let you know what your ethnic heritage is and other items in your genome. So a large number of 23andMe customers were involved, namely 231,000. So you can see there is an inflation and explosion of the number of subjects that are being used in these studies. We started with 30,000, then we went to 100,000, now we're at 231,000. And here the study makes reference to their inability to repeat the finding that was seen in the Han Chinese population of the two risk factors, most importantly the Sirt1 gene. Now this could very well be due to the different frequency of these alleles in different ethnic populations. So here then is the Manhattan plot of this newest study of 15 loci identified to be linked to major depression. And there is a number here involved. We will focus on this here for a moment. And so this data set then points to genes involved in transcription factors, coding for transcription factors, which are known in CNS development functions. For example, the MIES2 regulated pathway is involved in neurogenesis, together with other genes as well. So then, some of the problems that we encountered were sample size, heterogeneity of the illness, ethnic populations, and now we come to diagnosis. What kinds of symptoms do we count? How do we make the diagnosis of depression? And psychiatrists are boxed in pretty much by the Dsm-5 in its current edition. And we are basically bound to it, because if you want to file an insurance claim for a patient that you saw, you need to quote the Dsm-5 code by which you diagnose the patient with major depression. And the Dsm-5 has certain criteria which have to be met in order for the diagnosis of major depression to be made. So here now is a study that analyzed what are good depression symptoms to make a diagnosis and which ones are bad. And it looked at the centrality of DSM and non-DSM symptoms in depression by using something called network analysis. Which symptoms are connected to each other? And I'll show you the basic summary, namely that from a network perspective, the standard psychometric notion of depression is not really supported by the outcome of this study. Non-DSM depressive symptoms were just as important in terms of network analysis as were the classical DSM-related symptoms. And you see this here. For example, here is a cluster of symptoms that are separated from the network as a whole. Here is the strength of the association of the diagnosis with various symptoms. Energy loss and sadness are classics, of course, for the DSM, but here we have sympathetic arousal, which doesn't make any appearance in the DSM, but rates very high in the network analysis. We also have panic and phobia, which is totally not involved in the DSM for major depression, but is a very highly linked symptom in network analysis. Also, anxiety and tenseness here makes an appearance, and so on. Here is another analysis which looks only as the 15 IDS DSM symptoms, and you can see the line again, the correlation or the linkage, and energy loss and loss of interest, of course, here are on top. But self-blame, retardation are also important. Here is another one, concentration and problem-solving ability is also decreased. So here then is the upshot. We have formally talked about depression and our inability to carve depression by its joints, to carve nature by its joints, to look for those components that truly causally hang together. And the DSM and our classical approach has failed quite miserably. There is a movement now to do either genetic studies or imaging studies to come up with more tightly linked diagnostic criteria. It is very unlikely that the psychiatry profession will make any changes in the DSM-5, as huge amounts of effort and money have now been involved in this instrument. Every 2 or 3 or 4 or 5 years, a new edition of the DSM comes out, is being reprinted, resold. Psychiatrists are retrained in the new criteria. It's an industry. And it's very difficult to shut down an industry, even though its validity in terms of carving nature at the joints, in terms of depression, is in grave doubt. Now, we will include this talk into our module that we have just created, Genetics in Psychiatry, which is on our website that you can access together with talks on schizophrenia. We have also talked about genetics in Alzheimer's disease, and you can look at those studies in our module on the neurodegeneration project. Thank you very much for your attention, and we'll see you soon again for a presentation here at Behavioral Health 2000.